Hello everyone. So today we're going to be looking at lipids and membranes. So we're going to look at the structure of lipids, structure of cell membranes, and the function of these membranes. So another reminder, we'll see this quite a lot. We're kind of using this to guide us through the lectures. So again, what are the essential features of cell? They can store and transmit information. So that's that's what we looked at in our last couple lectures with our transcription, translation, and proteins. So if you want to check that out, you can. And 2, cells have a plasma membrane. So, through the structure of lipids and proteins that dictate the structure and function of the biological membranes. So, that is how we get this plasma membrane. So, what is the structure of lipids? So, here are a bunch of different examples of a few lipids. So, here's our triglycerides. Here are steroids, which are in many different molecules, and our fatty acids. So, here's a palmitic acid. We have our hydrocarbon chain and our carboxyl group. Here we can see in triacylglycerol or triglycerides, they're kind of commonly referred as either one, but this is the correct technical term. We can see here fats are formed by the addition of 3 fatty acid chains to the glycerol. So here are 3 fatty acid chains. So all lipids have hydrocarbon tails. These are the CH2. So here we can see there. See here, CH2, CH2, CH2. All lipids are hydrophobic, so they don't want to be with water. And lipids are a chemically diverse group with multiple functions. So lipids, as we'll see, are involved in many different things. So again, in their steroids, so all of our steroid hormones and external steroids that we can take. We have our triglycerides, which can be used for energy as well as be in food. Then we have our fatty acids. So certain ones we have like omega-3 and omega-6 fatty acids. So the structure of lipids. So the major lipid found in our cell membranes are the phospholipids. That's why you've probably heard it referred to as the phospholipid bilayer, because it's created from these phospholipids, which is this entire molecule right here. It essentially is a modified triglyceride. So the phospholipid has 2 regions. We have a hydrophilic region that's associated with water. So this is on the outer membrane, and this has a polar head that's formed of glycerol, phosphate, and a polar group. So here's that glycerol, phosphate, and a polar group. Again, phospholipids have a hydrophilic and hydrophobic components. So the bottom part here, we have a hydrophobic region, which has 2 fatty acid tails, which are these tails here. So this is kind of 3 representations of the same molecule. Again, phospholipids can be represented in a variety of ways to emphasize the overall structure in different domains in 3D shape. Again, so phospholipids are considered amphipathic, which means they're both hydrophobic and hydrophilic. So if you picture this as a cell, so here would be the inside of the cell. The inside would be hydrophobic, while the outside is hydrophilic, essentially creating that barrier. So other lipids found in the cell membranes are cholesterols. So you guys have definitely heard of cholesterols before, but with food and diet, but it's also an essential component of our cell membranes. So if we had no cholesterol, that'd be bad. So we still actually need cholesterol from our diet. So here is our cholesterol molecule. So cholesterol is considered a steroid. It is also amphipathic, and it can decrease our fluid. Cholesterols can increase or decrease the fluidity of the cell membrane depending on the temperature, which is very important. So at a normal cell temperature, the interaction of a rigid structure of cholesterol with the phospholipid fatty acid tails reduces the mobility of the phospholipids and fluidity of the membrane. So reduces. At lower temperature, so during the cold, cholesterol prevents phospholipids from packing tightly and increases membrane fluidity. So the amphipathic structure of cholesterol allows it to pack tightly with phospholipids. So you can see it's within the cell membrane. So again, cholesterol provide fluidity to membranes. That's very important. Cholesterols are crucial for function due to the lipids. Phospholipids can move laterally within a layer. So here we can see a bunch of different examples. of its movements. You see a bunch of different variations of the cell membrane. The type of hydrocarbon tails in the phospholipids can be either unsaturated or saturated. So here we have saturated versus unsaturated hydrocarbon tails with kinks, while the saturated hydrocarbon tails. So you can see that here we have unsaturated, we have those little kinks. So steroid cholesterol has different effects on membrane fluidity at different temperatures, like we said. So what are these structures of the membranes? So the goal of these membranes are to separate the cell's interior from the external environment and to enclose cells and organelles inside, and is also responsible for selective permeability. So we don't want to let anything get into the cell. We want to be selective with what we need. So here's an example of a plasma membrane. So here's our layer. Again, here's a chloroplast, and here's a mitochondrial membrane. As we'll talk about later, there's 2 mitochondrial membranes, but we'll discuss that later. So, when describing the structures of membranes, we can discuss it in the fluid mosaic model. So, in this, lipids and proteins coexist in a membrane forming a mosaic. The molecules have moved laterally in the membrane, so the membrane is fluid. So here we can see all different types of receptors and different proteins entering and leaving the membrane. So here's the phospholipid bilayer and outside the cell. Here's the inside of the cell. So you can see here we have a phospholipid bilayer, we have our cholesterol molecules, we have glycolipids, we have glycoprotein, we have integral proteins and peripheral proteins. So as we said, these membranes are full of proteins, either receptors or proteins anchored to the cell. So here we can see integral membrane proteins, which include transmembrane proteins that span the entire membrane. So here we can see they go from the top to the bottom. We also have peripheral membrane proteins, which are temporarily associated with either the internal or external side of the membrane. So it can be initially down here and then move up here, or vice versa. So membrane proteins. So membrane proteins are asymmetrically distributed in the membrane. They can move freely within the bilayer or can be anchored to a specific region, like lipid rafts. So we saw earlier that they can be fluid and move around, but some are anchored as well. So this includes different kinds and numbers of vary among different membranes. So membranes of certain cells will have different receptors, as we talked about quite a bit in immunology. So there's different types of protein. We have our peripheral ones, which are on one side of the membrane, and integral ones, which are transmembrane, which span the entire membrane, which we discuss up here, integral membrane proteins. So as we talked about, these proteins can function as many different things. They can function as a channel, a carrier protein. So these are transporters. So these can freely enter. These need to be pulled in through using energy in most cases, or put out. We have receptors, so our steroid hormone binding to a receptor, causing transcription. We have enzymes, and we also have anchors. So the differences in membrane structure lead to a variety of functions. So again, we have receptors, which allow cells to receive signals from the environment. So this can be from cytokines, hormones, a bunch of different things. We also have our enzymes, which catalyze chemical reactions, which help us in pretty much most of our important reactions. Then we have anchors. So anchors attach to other proteins that help maintain cell structure and shape. And then we also have transporters, which move ions or molecules across the membrane. So these include channels, carriers, and pumps. And we'll talk about these different pumps and stuff between this course and biochemistry. So what is the role of membranes in transport? So both lipids and proteins play an important role in controlling the movement of materials into and out of cells. The membranes are again responsible for selective permeability. So there's several different types of transport, but the main ones are passive transport, means no energy required, which is most times we use energy, it's ATP. As we saw in DNA, we use GTP, but in most cases ATP. So types of passive transport include simple diffusion, osmosis, and facilitated diffusion. So these are very important. You'll see these in a bunch of different topics in physiology, where calcium ions, potassium ions, sodium ions in neurology. For action potentials, this stuff is really important, fundamentals for all of those processes. So I recommend learning these, especially for physiology and stuff like that. So another type is active transport. So passive versus active is that it requires energy. Energy is being used to move something across the membrane, either in or out. So there's primary active transport and secondary active transport. So primary active transport is when we're using energy to move this molecule. That is the whole thing. Secondary active transport is when we're using energy for another molecule. And then this also one will come with it, using the piggybacking essentially on that energy. But we'll discuss this in more detail. So passive transport. So simple diffusion is essentially just the movement of solute molecules across membranes. So essentially, they usually work on gradients from high to low. So here we got a high to low. They can move freely. They can essentially just move freely through the membrane, but based on concentration gradients in most cases. This is common with small non-polar lipid-soluble molecules. So important is lipid-soluble, which means it can pass through the cell membrane, the phospholipid bilayer. So here we have a high solute and lower solute on each side, high, low. There's a net movement of solute from an area of higher to an area of lower solute concentration. So when we have equal complete permeability and we have equal on each side, there will be no net movement because we always move from that high to low. So net movement of lipid-soluble solutes and molecules is unidirectional. Solutes move from an area of higher concentration to an area of lower concentration down a concentration gradient. So this is super important, especially in physiology. So we'll talk about gases and pressure and blood. All of this stuff works on pressure gradients, which is super important. It's also related to a lot of physics, but again, this is a key fundamental. So these passive transports from high to low work until this dynamic equilibrium is reached. So essentially, these will move. Once there's equilibrium, we'll have no movement. Unless something, so like for example, in the circulatory system, this won't happen because we have something that keeps increasing the pressure, like our heart. So it keeps that movement going. So another form of passive transport is osmosis. So you may have heard of osmosis in science classes or even the cartoon Osmosis Jones. I'm dating myself there. But anyways, this involves the movement of solvent molecules across membranes and again involves water. So here we have our water and solute and we have osmosis. So high solute concentration with lower water and a lower solute concentration with high water. Again, membrane allows passage. So here, equal solute and water concentration. So the goal is to have that balance of concentration and solute. So here, there's a net movement of water molecules is unidirectional once again. The solvent moves from an area of higher concentration to an area of lower concentration down a concentration gradient. This occurs until dynamic equilibrium is reached, and for both water and the solute concentrations. So we can see here that the space is also important. They space themselves out as well. So that occurs with the solute. So again, water will move passively from hypotonic solutions. So solutions that are hypotonic have high H2O. Tonic kind of refers to the solute. Hypertonic solutions have low H2O. So animal cells. So in animal cells that lack cell walls, such as red blood cells, these cells must remain isotonic relative to the external environment. Because again, these are circulating through the blood. So in plant cells, which have cell walls, cells must remain hypertonic relative to the external hypotonic solution in the environment. So you can see here, there's a good example of how human cells and plant cells can differ. So here's an example of a hypertonic red blood cell. So here we can see the blood, if the water is removed, our shell becomes all shriveled up. In our isotonic, where we have balance, our cells remain normal. If we have hypotonic, where we have too much H2O, our cells can swell and burst. And again, that is not good. So another form of passive transport is our facilitated diffusion. So facilitated diffusion involves the net movement of solutes, including ions, small molecules, down concentration gradients until equilibrium is reached. Both polar and non-lipid soluble solutes do not pass through the membrane on their own. They require help. So again, facilitated. They're facilitated by something. They need help. These help come through proteins in the membrane. So these types of helper proteins include channel proteins and carrier proteins. These are both integral transmembrane proteins. So remember, we have our phospholipid bilayer. Bad drawing. I'm just going to draw a few of these. And we have a protein that can pass through. So again, a channel protein is kind of something that just passes through, or we can have carrier proteins. So here's an example of several different mechanisms. So we can have simple diffusion, which is simply this molecule is able to cross the plasma membrane on its own fruition. So it can just go through. We have channel mediated, so these can sometimes be opened or closed. So example, in neurophysiology, something can stimulate the muscle to stretch. This can open, and we can allow stuff to go through. So this is channel mediated. And then we have carrier mediated. So again, they're taken in by a protein and then transported into the cell. And again, we have our concentration gradient, and usually of high to low. So these ones we need high to low because again, it's passive. There's no energy. So now moving on to active transport. So active transport, again, the key thing here is requires energy to move substances across a membrane against a concentration gradient. So now we're going from low to high. So solutes such as ions and molecules move from an area of lower concentration to an area of higher concentration. So in passive active transport, we need energy supplied by ATP, like we mentioned, and transporter proteins. So there's several specialized transporter proteins we'll talk about in physiology. So one's for thionine, thiodine, or in thyroid stimulating hormone. A bunch of different hormones have different transporter receptors and stuff like that. So there's a bunch. So again, there's primary, which uses energy directly, and secondary, which uses energy indirectly. So primary active transport uses energy directly. Energy from ATP hydrolysis, so that's when we go from ATP to ADP, releasing those phosphates, is used to pump ions into or out of cells against the concentration gradient. Only cations are transported by pumps in primary active transport. So a really common example is the removal of sodium and internalization of potassium. So this happens in a lot of muscle contractions and stuff like that. So again, we have higher sodium out here, high to low, but through these proteins using energy, here we go from ATP to ADP, we can take in 3 sodiums and take in 2 potassiums. So that's a common thing we'll talk about when we talk about neuroscience, but we can release 3 sodium and take in 2 potassium ion. But anyways, this keeps the gradient either way, but we can see that this is the example of primary active transport. We're using energy to move sodium out and potassium in. So in secondary active transport, we use energy indirectly. So here we can see we have active transporter pumps protons across the cell membrane using ATP directly. Again, this is using primary active transport. The pump, this creates a proton concentration or chemical gradient and electrical gradients, since protons are charged that can do work. The movement of protons down their concentration gradient drives movement of other molecules against their gradients, again through secondary active transport. So for example, protons are pumped across the membranes by primary active transport. The proton pump generates this electrochemical gradient, and with the higher concentration of protons outside the cell and a lower concentration of protons inside the cell. So here we have an antiporter, we'll discuss later, uses the proton's electrical chemical gradient to move a different molecule out of the cell against the concentration gradient. For example, we have the hydrogen ion pump in animals and plant cells. So that's a very common pump in physiology. So summary, transport processes through membranes. So we have transport mechanisms. So simple diffusion, no energy, so it's passive. Driving force is a concentration gradient. No membrane protein required and is not specific. Facilitated diffusion, again, is passive. Uses a concentration gradient, but needs a protein, and it is specific. So only certain things can pass through certain proteins. And active transport, which requires energy, which is through the ATP hydrolysis, moving against the concentration gradient. Yes, they need proteins, and it is specific. So I hope you enjoyed that one. Again, this is really good fundamentals for both physiology and pretty much any field of medicine, because because you need to know these different transporters, because essentially everything has receptors and moves in and out of cell. But yeah, I hope you guys enjoyed it. See you next time.